Bismillahirrahmanirrahim. In this lecture, we'll discuss how can we classify the bacterial and the antibiotics in one-page approach. To start, we have some difference between the gram-positive and the gram-negative bacteria. The gram-positive bacteria appear in the blue colors, while the gram-negative bacteria appear in the pink colors in the gram stain. And this because the outer membrane of the gram-negative take the stains of the pink or red colors. Another difference that the gram-negative bacteria has what's called Bourne channels, and these Bourne channels we have some clinical implication in the when we need to understand the mechanism of resistance. And some examples of the gram-positive and the gram-negative bacteria, we have some aerobic infection gram-positive cocci. If the bacteria appears in clusters, this is called the Staphylococcus species, and if appear in the chains, we have the Streptococcus and the Enterococcus. And we have aerobic gram-negative cocci and bacilli. For gram-negative cocci in aerobic infection, we have some example like Neisseria species appear as a diplococci. And for gram-negative bacilli in aerobic infection, we have either bacilli like Enterobacterales or Pseudomonas or Coccobacilli like Acinetobacter. And we have other anaerobic infection like a gram-positive anaerobic bacilli like Clostridium species and the gram-positive anaerobic gram-negative anaerobic bacilli like Bacteroids. We have third type of bacteria like it's called atypical bacteria. The atypical bacteria have no cell wall, and because this bacteria has no cell wall, there is no gram staining. And there is and because there is no gram staining and it doesn't appear in a conventional cultural sensitivity. And example of these bacteria like Mycoplasma, Chlamydia, and Legionella. And the importance of this information that some types of infection may be due to atypical bacteria that will not appear in the conventional cultural sensitivity. For example, community-acquired pneumonia. So these types of bacteria need to be covered during empirical therapy. How can you classify the antibiotics? Actually, we have two types of classifications. The antibiotics can be classified according to the pharmacokinetics and the pharmacodynamics or according to the site of the action. And let's start with pharmacokinetics and pharmacodynamics. The pharmacokinetic and the pharmacodynamics, the antibacterial can be classified into bactericidal or bacteriostatics. And each types can be, and each classification can be sub-classified into time-dependent and concentration-dependent. So we have time-dependent bactericidal group, and we have time-dependent bacteriostatic group. And we have time-dependent, concentration-dependent bactericidal and concentration-dependent bacteriostatic. What's different between what's meant by time-dependent and concentration-dependent? If we draw this graph that shows the blood level of the antibiotics during single injection of the drugs, the antibiotic level increased in the blood level, then it fades again till it reaches the baseline. And this line represents what's called minimum inhibitory concentration. What's meant by minimum inhibitory concentration? The minimum inhibitory concentration is the minimum concentration of the antibiotics that inhibit the growth of the bacteria. So if the antibiotic blood level increased above the MIC, the bacterial growth will be inhibited. And based on this information, we have two types of the antibiotics. Either we have time-dependent or concentration-dependent. Time-dependent antibiotics, these group of antibiotics, the goal is to keep the drug above the MIC as long as possible. That's why continuous infusion of these drugs may be recommended in some cases. And for example, beta-lactam antibiotics like piperacillin, tazobactam, or meropenem. On the other hand, the concentration-dependent antibiotics, the goal is to keep the peak level of the drug as high as possible. So these type of drugs should be given as once or twice daily dose. For example, aminoglycoside. And the second classification of the antibiotics is classified are based on the site of action. We have group of antibiotics that inhibit the cell wall, like beta-lactam antibiotics and glycopeptide. And we have another group that inhibit the cell membrane, like polymyxin and daptomycin. We have groups of antibiotics that inhibit the protein synthesis of bacteria, like aminoglycosyl dinesylate, like cyclin macrolide, and clindamycin. And finally, we have a group of antibiotics that inhibit the DNA synthesis, like fluoroquinolones and the metronidazole. And in the coming lectures, we will discuss each group separately. Thank you.